The promoters and operators of bacterial genes and operons bind regulatory proteins that can activate or repress transcription. Artificial gene constructs called reporter genes can be used to identify the proteins that regulate bacterial genes. Let's see how. Suppose we want to identify the proteins that bind to the regulatory region, the promoter and operator of a gene. We can use molecular biology techniques to fuse that gene's regulatory region to the coding sequences of another gene, in this case LAC-Z. Now we have a reporter gene. The fusion gene will report to us whether or not the gene's regulatory sequences are activating transcription. When the reporter gene is transcribed, beta-galactosidase protein is made. The LAC-Z gene is used in reporter gene constructs because it's easy to tell whether or not a bacterial colony is making beta-galactosidase just by looking at it. The normal substrate of beta-galactosidase is the sugar lactose, which beta-galactosidase breaks down into galactose and glucose. However, beta-galactosidase can also use this molecule called X-gal as a substrate. Beta-galactosidase breaks down X-gal into galactose and another molecule that oxidizes to produce a blue precipitate. If you add X-gal to the medium in a Petri plate, bacteria that make beta-galactosidase will accumulate the blue precipitate. The colonies will be blue. Bacteria that don't make beta-galactosidase will form white colonies. To use the reporter gene, you want the LAC-Z gene in the reporter to be the only source of beta-galactosidase. To achieve this, you use a bacterial strain where the LAC-Z gene in its chromosome has an inactivating mutation. You transform this mutant strain with a plasmid containing the reporter gene. If you grow the transformants on medium that has X-gal, blue colonies indicate that the reporter is being transcribed, and white colonies indicate that the reporter is not being transcribed. Now let's see how we can use this system to identify transcriptional regulatory proteins. Suppose you've discovered an E. coli gene that is transcribed only in the presence of the sugar maltose. You could make a reporter gene that will help you to identify the regulatory proteins that mediate the response to maltose. When LAC-Z minus bacteria are transformed with the reporter and plated on medium containing maltose and X-gal, the colonies should be blue. When plated on medium with no maltose, the colonies should be white. How can we explain the maltose responsiveness of this gene's regulatory region? One possibility is that an allosteric repressor protein exists, analogous to the lactose repressor. In the absence of maltose, the repressor binds to the gene's operator and blocks transcription. When bound to maltose, the repressor cannot bind the operator, and so the gene is transcribed. If the negative control hypothesis is correct, in a strain with a loss-of-function mutation in the gene that encodes the repressor, no repressor will be made, and so the reporter gene is transcribed constitutively. Beta-galactosidase is made and colonies are blue whether maltose is present in the medium or not. To screen for such a mutant strain, you can grow LAC-Z minus bacteria in the presence of a mutagen, transform the bacteria with your reporter gene, and plate the cells in the medium with X-gal but no maltose. Almost all of the colonies will be white because in the absence of maltose, transcription from the reporter will be repressed. A rare blue colony should have a mutation in the gene encoding the repressor, so the reporter is transcribed in the absence of maltose. The repressor gene can be identified by transforming the mutant cells with a wild-type E. coli genomic library. The cells are plated on medium with a drug to select for transformants with a library plasmid, X-gal, and no maltose. A rare white colony will have been transformed with a library plasmid that contains a wild-type copy of the repressor gene. Because the repressor is made in those cells, the reporter in that colony is not transcribed in the absence of maltose. Most of the colonies will be blue because they are transformed with plasmids containing other E. coli genes that don't make repressor. If the negative control hypothesis had been incorrect, you would not have found mutants in your screen in the first place. In that case, another possibility is that the maltose-responsive gene is controlled by a positively acting protein, an allosteric activator that can bind the promoter only when bound to maltose. In the absence of maltose, the reporter is not transcribed because the activator can't bind the promoter. If the positive control hypothesis is correct, in a strain with a loss-of-function mutation in the gene that encodes the activator, no activator is made, and so the reporter is off even in the presence of maltose. The colonies are white whether maltose is present in the medium or not. To screen for mutations in the activator gene, you can grow LAC-Z minus bacteria in the presence of a mutagen, transform the bacteria with your reporter gene, and plate the cells in a medium that contains maltose. Almost all of the colonies will be blue because in the presence of maltose, transcription from the reporter will be activated. A rare white colony should have a mutation in the activator gene, so the reporter is not transcribed even in the presence of maltose. The activator gene can be identified by transforming the mutant cells with a wild-type E. coli genomic library. The cells are plated on medium with a drug to select for transformance with the library plasmid, xgal, and maltose. A rare blue colony will have been transformed with a library plasmid that contains a wild-type copy of the activator gene. Because the activator is made in those cells, the reporter in that colony is transcribed in the presence of maltose. Most of the colonies will be white because they are transformed with plasmids containing other E. coli genes that don't make activator.